the stem cells of these two bilaminar embryonic disc. Bilaminar embryonic disc, the flat disc. You just can imagine for the pancake or the pizza bread, as if the two pizza breads are there, one after another. Similarly, the two embryonic discs, epiblast and hypoblast, they are present. Okay, the stem cells of these two layers, bilaminar disc, they migrate in different way to form the different shape and morphology of the embryo. That's why this cellular movement is also known as morphogenic movement. So simply we can say gastrulation can also be called as morphogenic movement. That how it starts? That gastrulation starts with the formation of primitive streak. Suppose this is the epiblast. This is the epiblast. Suppose this is the cephalic end, means head region. This is the tail region. Then how the primitive streak is formed? From the caudal region towards the cephalic region, the primitive streak is formed and a furrow or a line is formed. Then towards the anterior end, suppose this is the cephalic region, to which side the head is present. Towards the anterior end of the primitive streak, it is swollen to give rise the primitive node. Primitive node, a mass of epiblast cells. Here are the epiblast cells. It consisting a primitive pit. A primitive pit is formed. Then this primitive pit, the depression, shallow depression. What is this? It is a shallow depression. It is a shallow depression. Not so deep, shallow. Depression. Then this primitive pit is extended towards the caudal region along the axis of the embryo, extended towards the caudal region, and it forms when it is extended, it forms the primitive groove. Groove, a longitudinal depression. Then this is the epiblast. What is this? It is the epiblast. It is the head region, cephalic region, cephalic end. This is the caudal end, tail region, caudal end. Then what happens when this groove is formed? The cells of the two sides. When the primitive streak is formed, it establishes the cephalic. Suppose this is cephalic region and this one is caudal region, like this. When the primitive streak is formed, then the cephalic and caudal region are established. Even the left and right sides are also established. Left hand, suppose this is left, this is right, sides are also established. As the primitive groove is formed, then the cells of the epiblast from both the side, they migrate into the primitive groove. That begins the morphogenic movement of the cell. Below the epiblast, the hypoblast is here. The two embryonic discs, how they are present, suppose the epiblast consists of the columnar cells, suppose after this the trophoblast cells are at us. Suppose this is the cephalic margin, after that what is that? Trophoblast are there. These are trophoblasts. Suppose here this side is the caudal margin. Same after the trophoblasts are there. Here the primitive streak is suppose formed in the middle of the epiblast. Now let us get what happens. As we have studied the bilaminar disc when formed, then the hypoblasts, they proliferate and differentiate to form the yolk sac, primary yolk sac, that is the one of the extra embryonic membrane, rudimentary form of the extra embryonic membrane, that is yolk sac, and other is formed the amniotic cavity. Suppose this is these are the columnar cells of the epiblast, and here are the cuboidal cells of the hypoblast. Which form the primitive endoderm. What it forms? The cells of the hypoblast, they form the primitive endoderm which forms the extra embryonic membrane. As I told you, when the primitive streak is formed, then what happens? The side cells, the cells from the side of the epiblast. Of course, the cells of the epiblast from the sides, they migrate and enter into the primitive group. First they proliferate, they divide. They divide to produce many. Here they divide. You can say here they divide many cells of the epiblast. They divide. Then they migrate. They move into the primitive. We can make it more larger here. A primitive group. Okay. Here the cells of the epiblast, they divide and move into the primitive group. Move into the primitive group. So they enter like this. And make a layer of flask shaped. Then they detach. The inward movement of these cells, the proliferated cells, the migration of proliferated cells, proliferation, the cell division, migration movement, when these cells are produced by the cell division from these epiblast cells, and because all these are stem cells, and they migrate into the primitive groove, and then they detach and slip down the epiblast. The primitive endoderm is replaced by these newly formed cells. Inward movement is called invagination and detachment is called delamination. So these are the different form of the movement of the cell, mass of cell or sheet of cells. Now this is definitive, the final, this is the primary endoderm or definitive or primary endoderm. The previous endoderm which was formed by the hypoblast, that is the primitive endoderm which forms the extra-embryonic membrane only. But the embryonic tissue are derived from the definitive or primary endoderm. Now, all cells are not required to replace the primitive endoderm. Then some cells remain over here and still they are differentiated, and cells are proliferated from the caudal margin. Some are remain here in the space in between the epiblast and hypoblast. Now no more hypoblast, newly formed endoderm. The cells are proliferated from the caudal region and remain in between the epiblast and newly formed endoderm. Newly formed endoderm. This layer forms the mesoderm. The space in between the epiblast and the endoderm filled with newly formed proliferated cells that are delaminated from the epiblast as well as proliferated from the caudal margin. They form the another layer, the mesoderm. Primary primary mesoderm. The rest of the cells left in the epiblast. That makes the proper embryonic disc. I have forgotten to tell you one thing. This epiblast is the proper embryonic disc because that from that epiblast only all the three germ layers are developed. So this is called proper embryonic disc, and this is nothing but the epiblast, and the rest of the cells which are left in the epiblast or the proper embryonic disc, that becomes the ectoderm. Now this becomes ectoderm. Friend, these three germ layers are formed. Ectoderm, mesoderm and endoderm are established. There is no, previously the cells are moving into the primitive group. Now there is no cellular movement. Let us look into the animation of gastrulation. Towards the end of the second week of development, the bilaminar embryonic disc consisting of hypoblast and epiblast has formed. Throughout the third week of development, the cells of this embryonic disc undergo proliferation, differentiate, and migrate and rearrange to form the three germ layers by a process called gastrulation. After 15 days of fertilization, another structure is formed, primitive streak towards the caudal end. The primitive streak defines the major axis of the embryo, such as cranial end towards the head, cranial or cephalic end, caudal end towards the tail, as well as left side and right side of the embryo. The primitive streak towards its anterior enlarges to form the primitive node made up of cells of the epiblast. It consists of a depression called primitive pit, and this depression extends towards the caudal end, forming the primitive groove. After the formation of primitive groove, the cells of the epiblast from both the sides migrate into the primitive groove by a process called invagination. These cells then detach and slip beneath the epiblast, and they replace the primitive endoderm, replace the cells of the primitive endoderm to form the definitive or the primary germ layer, that is endoderm, primary endoderm. After 16 days of fertilization, the rest of the cells which are present on the epiblast form the ectoderm, and the cells, newly formed cells which are present in between the epiblast and newly formed endoderm, they form the another germ layer, mesoderm. After the formation of these three germ layers, ectoderm, mesoderm and endoderm, there is no more migration of the cells of the epiblast into the primitive groove. Thus, these cells, from these precursor cells, the different embryonic tissue are developed.